Hello, judges. I'm Yuxuan Zhang, and today I'm going to present my project, Lichens as Bioindicators of Air Pollution. So first, some background research on what lichen is. So they're a complex organism and is a symbiotic relationship of a fungus and an alga. So the fungus provides the appearance of the lichen, and the alga provides the nutrients. So here's an example of the structure and the appearance of lichens. And because they're a long-lived organism, meaning that you can monitor them year-long and they're widespread across all continents, they are very important as a bioindicator. And moreover, they get their food source directly from the air, meaning that pollutants can accumulate very quickly. So there are two types of pollutants lichens are indicators of. The first one is nitrogen oxide, which is produced from car exhaust and fertilizers. Different species have different varying tolerance towards that, and the presence and absence indicate the amount of nitrogen in the atmosphere. And another pollution is ethylene, which is actually naturally occurring during fruit production, fruit ripening, but when it's released in large quantities from traffic and industry, it actually causes a reduction of flower and fruit production. So my scientific question is, how does changing the pollution in the atmosphere affect the growth of lichens? And my hypothesis is that if pollutants are added to lichens' growing environments, then its growth will be inhibited and its health will be damaged. And this is a very important question to consider because lichens can help scientists to gauge the pollution in the environment without using expensive equipment. So part one of my procedure was that I went to four different local locations shown here and observed the sizes, different species in those areas. So here's an example of the samples I collected. So part two was the actual experiment. So I took home the samples and I divided them into three groups. Car exhaust, which I collected from the car pipe and used plastic bags to seal the samples in it for about 1 hour, and ethylene, which I collected from banana and control group. So I took data on the areas, mass, and physical traits of the samples. So some notable physical changes I saw was that, first of all, chlorophyll degradation, meaning that there was a lack of greenness. You can see that the sample become browner and paler. And abscission, which is like parts of the plant has fallen off. Moreover, I saw an increase in fungi. So here you can see that there was a buildup of fungi. And moving on to my data analysis, for a control group, and I actually saw that for Jamaica Pond and Blue Hills, there was an increase in change of area, and for Lars Anderson and Sensor Street, the change in area was negative. So this might mean that samples might have been previously exposed to pollution, so 2 months was not sufficient for them to recover. So continuing, we have the car exhaust group. So this group actually shows a trend of decrease in area, which proves my hypothesis. And moreover, you can see from this picture that there are less lichens on the substrate at the end. And for ethylene group, there were some unexpected results, which actually shows an increase in area. And this means that ethylene actually promotes the growth of lichens, similar to its food ripening effect. So I also took some data on the mass, which was the method two, but it was inaccurate. So in conclusion, my hypothesis was partially supported, and car exhaust decreased the lichens coverage, but ethylene promoted the growth. So we should be alarmed by a decrease in lichen locally from the car exhaust experiment. And some errors that were involved was that the scale could not collect accurate mass data, and it could not represent the diversified population. And some limitations were that I cannot conduct this in natural environments since it's winter, and there were no equipments for me to detect the types of pollutants in the lichens, which can be very helpful to support my results. And here's my works cited, and thank you for listening.